This Week in Virology, the podcast about viruses, the kind that make you sick. Hello, Berkeley. All right, not bad. It's time for This Week in Virology, episode number 228. Today is April 13th. Today is April 13th, 2013. Hi, everybody. I'm Vincent Racaniello, and this is TWiV, the podcast all about viruses. Today, we're coming to you live from Berkeley, California, at the University of California, and this is the Microbiology Student Symposium. And I have a couple of great faculty from the university joining me today. The first is a professor in the Department of Plant and Microbial Biology, Britt Glaunsinger. Welcome.
Thank you.
Thanks for joining us today.
Thanks for having me.
Our other guest is a professor in the Division of Infectious Diseases and Vaccinology, Eva Harris.
Hello.
Did I get the affiliation right?
Mm-hmm.
All right, thank you. Now, before we go on, what do we have to do? Yeah, the weather. It's really nice here. When I left New York on Friday, it was raining. Let's see what it is. It's sunny, blue skies, puffy clouds. Let's see, 22 degrees Celsius. It's like that every day, right? More or less. So there you go. That's the weather, so we don't forget that. So I wanted to talk with these two wonderful virologists about the work they do. The cool thing about doing a podcast at a meeting is that you can get some of the people that are there and expand the reach that we have. And usually we do this in my office. Every Friday I connect to people via Skype and record. But now and then it's good to get out into the world. So I appreciate the opportunity to do this. Let me see before we go on. How many TWiV listeners are there here? Not bad. Almost like 80%. Of the 10,000 people who are here. It's good, you're good fans. Let's start with you, Britt. Now, the real hard question for you is, do you know the commonality in our two careers? It's very obscure. It's very obscure.
Boy, so I'm going to guess it's not a passion for viruses.
Well, that would be a good answer. No, just the virus you work on was discovered at Columbia.
Yeah, sure.
Would you agree?
That's right.
That's right. So tell us about your background. Where did you get where were you born in the U.S.?
I was born in Arizona.
Okay.
And so this is officially the coldest place I've ever lived, here in Northern California. It's pretty rough, I must say.
Where did you get your PhD?
I got my PhD at Baylor College of Medicine in Houston, Texas, in a department of virology at the time. So I've been a virologist and in love with viruses for a while.
That department is full of virologists.
It sure is.
Yeah. And your postdoc?
My postdoc was at UCSF, University of California, San Francisco, with Don Ghanem.
And that must be where you started working on KSHV.
That is indeed.
You don't like to call it KSHV? Or
no, that's what I call it,
KSHV. Which name do you prefer?
I prefer KSHV.
Kaposi sarcoma herpesvirus, right?
Yep, Kaposi sarcoma-associated herpesvirus.
Okay. So let's talk. I want to talk about a specific aspect of your lab's work, but first, we haven't talked too much about KSHV on TWiV. We did have Pat Moore on, but he talked about Merkel's virus. So let's get a little background on KSHV. I know it because it was discovered in the context of AIDS, right?
Yep.
Right, but clarify something for me. Before AIDS, there were old men in the Mediterranean who'd had Kaposi sarcoma. Mm-hmm. So did they just have KSHV or do they have AIDS as well?
No, these are AIDS-independent cases. Okay. You're right that there's definitely a strong link to HIV infection, and that's when people started taking notice of KSHV, or that's when it was discovered, was association with AIDS. But originally it was this disease of elderly Mediterranean men, and there's still more of a link to men than to women, and that's something that kind of remains a mystery. But the incidence of KS rose dramatically in the 1980s with the rise of sort of the AIDS epidemic, and so that's when it really started getting noticed.
Yeah, that was one of the case definitions that the CDC originally put out, KS or opportunistic infections. This was really KS or Kaposi sarcoma is really the AIDS-defining illness of the 1980s. That's what people picture, these men with sort of skin lesions, usually homosexual men at the time. So we don't know if they were genetically predisposed or immunosuppressed, those pre-AIDS people?
Yeah, so presumably there's some other factor, environmental factor, or genetic factor that's predisposing them to KS. That, at this point in time, is unknown.
Okay, so now today it's common in people with AIDS, but is it also found in people without AIDS still?
It can be found in people without AIDS, so it's very common in sub-Saharan Africa, for example, although there's also a lot of AIDS there as well. But in the Western world, it's thought to be about 6% of people have it, so 6% seroprevalence, and you don't necessarily have to have HIV to have KSHV, although that's definitely a risk factor because it's an STD.
Oh, so it's a sexually transmitted disease, also intravenous drug use,
yes.
perhaps?
Yes.
Okay. And if you are infected, I presume you can be infected anytime in your life from when you're sexually active or if you use drugs before that.
Yeah.
What happens when you receive the virus?
Well, for most of us, if we receive the virus, like if we receive most herpes viruses, so probably some listeners know and some don't, that all of us are infected with multiple herpes viruses, will be for our entire lives and
5?
have been.
Is that the number 5?
Yeah, probably. You've probably got at least 2 or 3, and that often makes people nervous, especially because they tend to think of herpes viruses as sexually transmitted diseases or causing cold sores. And truth be told, most of us have, those are simplex viruses, most of us have one of those too. Even if you don't get cold sores, you'll get it in, like a huge percentage of the population, and you can secrete virus without ever getting a cold sore, for example. So they're very efficiently spread. KSHV is much lower seroprevalence than some of those, but back to your question, when most of us get a herpes virus infection, our immune system does a remarkable job of taking care of it. And so we generally don't show any symptoms. Really, the problems come around when your immune system starts to fail, like in an AIDS patient.
Okay, so if you have AIDS, you get a sarcoma on the skin. Are there other kinds of cancers that are associated with it in AIDS patients? Or is that it?
So there are a couple of other types of B-cell cancers, so pleural effusion lymphoma, multicentric Castleman's disease. So yeah, KS is one of sort of the 3 types of cancer-like syndromes that KSHV can cause.
But in an immunocompetent host, usually there's no
immunocompetent host. Nope, the virus is hiding out in your B cells, so it will sort of undergo what's called a latent infection, where there's a very small amount of viral genes that are expressed, and it's flying below the radar.
But there is a period, I guess, when you're first infected, when you have a lytic infection.
Presumably, yeah, sure.
And are there periodic reactivations when you make virus?
You assume that there's going to be periodic reactivations. I don't know to what extent that's known for KSHV in humans, but certainly for other herpes viruses, it's pretty clear there are periodic reactivations. Usually when you're stressed, right? Yeah. For those people who do get periodic cold sores, it can be UV stress or the stress of an exam or something else like that can cause a reactivation event.
Okay, so that gives me the good background to proceed. Now, any other questions about disease? By the way, you can ask a question, right? The audience can ask at some point if you'd like. We'll repeat it for you. You can just raise your hand. Usually we do this on Twitter, but I can't multitask right now, so I'm not going to do that. All right, so actually I wanted to talk to you about one aspect of their work that was similar to a talk we just heard, which is about nucleases during viral infections. And in your case, you're interested in this whole idea that one of the things viruses do to impair the host is to degrade messenger RNAs, right? Is that correct in assuming that's a major interest?
Yes, that is a major interest.
Like all of your last 6 papers are on this, so I presume it's a major interest. So I'm interested in this general strategy first. So KSHV, you can infect cells in culture and look at the degradation of host mRNAs, right? Do all the herpes viruses do that, or is that unique?
No, not all the herpes viruses, although not just KSHV. So herpes simplex viruses also do this. They use a different protein than KSHV does, but the general overall mechanism is pretty similar.
Okay, so I learned that one of the endonuclease that does this is SOX. Is that correct? SOX?
Yes.
What does that stand for?
Stands for the baseball team. It
Good.
It stands for shut off an exonuclease.
Okay, so I understand that it is it's got some other function besides being an exonuclease?
That's right. Like most viral proteins, it's got at least one other function. And the other function that we know about for it is that it helps promote presumably appropriate DNA replication, viral DNA replication in the nucleus. So these are nuclear replicating large double-stranded DNA viruses. And when they replicate, you often get these sort of complex branch structures that arise, and those branch structures need to be resolved, and it's thought that this protein helps resolve those structures.
Is it a DNase or more of a topo-like?
It is like a DNase, but it's a DNase in vitro. It may function sort of like a recombinase in cells.
So what you found is it's also an RNase.
That's right.
It's an exo?
Right. It's an endo. That's
RNase.
a very important distinction for us. Yeah, it's an endo.
But it was originally, in many of your papers, you talk about alkaline exonucleases.
Yes, that's right. So when you ask me what it stands for, the confusing part, right, is that it's named shut off an exonuclease. That's because of its in vitro activity on DNA, where if you mix it with DNA, or for that matter, RNA in vitro, it can degrade exonucleolytically. But in a cell, it cuts an RNA in the middle somewhere.
Okay. So is this an essential gene in the virus? If you take it out?
It's a pretty important gene in the virus, but that's in part because of its secondary role in DNA replication.
So can you separate the two roles?
You can separate the two roles, and that's enabled us to ask, why is this RNA degradation activity important? And we found that we can do that we use a mouse herpes virus to do that, because we can do in vivo studies with the murine herpes virus. And what those studies have told us is that it actually plays a really important role in a number of different aspects of the viral life cycle. So when these viruses infect, say, a mouse initially, you're going to have a lytic replication in the lung. And from there, though, the virus needs to traffic to lymph nodes and spleens, where it's going to access the B cell population in germinal centers and establish latency. And so without the ability to degrade RNA during an infection, the virus can't traffic very effectively and is pretty seriously impaired at the latency establishment stage. So it tells us there's some pretty important role that widespread RNA degradation is doing for the virus.
Do you know what that is?
I've got some ideas.
It's not just a general thing of having to take over the translational apparatus?
That's right. Well, that's what people think, right? When you think about host shutoff, which is a phenomenon that a lot of viruses do, and they can do it through widespread RNA degradation, which is what we study, but they can do it through a whole slew of other mechanisms. The thinking is that, well, they do it because you want to reallocate resources towards the virus. Because viruses, of course, are stealing host machinery a lot of times to replicate, or as a generalized immune evasion strategy, or to manipulate the host response. And so it could be, in theory, any of those. But one of the interesting things that we're finding is that this widespread degradation of host RNAs, it's not just host RNAs. This is a way that the virus broadly controls viral gene expression as well. So that was sort of an unexpected finding that we've had. So I think that's going to end up playing an important role too.
So are all host mRNAs degraded?
There's a subset of them that escape. One of the escapees that we're really interested in is interleukin-6. And interleukin-6 is a gene that encodes for a cytokine that's important for B cell survival. So if you remember what I told you a minute ago, B cells are the cells that these viruses reside in latently. And so without IL-6 around, the virally infected cells don't survive very well. So there's a sort of a vested interest that the virus has in upregulating this cytokine. As it turns out, this messenger RNA directly escapes cleavage because we found it has this RNA element that helps protect it from SOX.
So that kind of thing always amazes me in terms of evolution, right? So what came first? The virus must have been there, and then somehow it utilized, because IL-6 didn't evolve to accommodate infection, right? So somehow the virus has evolved to utilize this as part of its pathogenesis.
Yeah. I don't know. I think that, You're right. Who knows what came first?
We can't. You don't know, but it's a cool thought experiment.
Right,
that I think when you have these kinds of interactions, you need to think about it, because I think it helps bring out other experiments that you can do.
Yeah.
So do we know what the target sequence is? Because you said they cut somewhere in the middle. Is there something that it sees?
There is a target sequence, which actually that, if you think about it, is unexpected, because we know that this enzyme comes into a cell and targets the vast majority of cellular RNAs. So when you think about why, or how would a virus that wants to deplete most RNAs from a cell go about doing that, you would think there should be relatively low sequence specificity. It might be a more efficient way to do it by, if it wants to target messenger RNAs, maybe it binds the cap and then cuts anywhere in a position-dependent manner rather than a sequence-specific manner, right? So we were really anticipating that there was not going to be a directed sequence type of recognition element, so we were pretty surprised when we found, in fact, that there was a sequence element that could direct cleavage. And we haven't completely mapped what that is. We know some components of it, and we're really curious to know what the others are. It's larger than we might think it should be, given the breadth of targeting. So it's something we're really interested in.
But it's usually in the middle of the RNA, roughly?
Could be anywhere.
Could be, okay. So if you express various RNAs and add the enzyme, it cuts them all over the place?
Sure.
Just once?
Usually once or a small number of times. It's not like it's prevalent enough that you're getting cuts all over the place. You're cutting 1 or 2, maybe 3 times per RNA.
And why aren't the viral, well, you said actually before some viral RNAs are cut.
Yeah,
right? Because
probably.
that was reading your papers, you don't talk about that too much, probably because you're working on it now. And,
that's our next paper.
I wanted to know why the viral RNAs are not cut, but some of them are. But the ones that are not, do you understand why they're not cut? Do they have the IL-6-like element?
Yeah, so the viral escapees, are they escaping because they've got some sort of dominant protective feature, or because they're just lacking a targeting element? And right now, we think, for the most part, it's because they're lacking a targeting element. That doesn't necessarily mean that there are not viral examples of things that have dominant protective features, but the ones that we've identified so far are escaping because they lack that element.
So you also have worked on this idea that this SOX collaborates with a cellular degradative pathway?
Yes.
Can you tell us a little about that?
Yeah, so we heard a little while ago about this enzyme XRN2, and the one that we're interested in is sort of a similar one called XRN1. And
Are they homologous, or?
They're similar in that they're both 5' to 3' exonucleases. So it's important to think about the difference between an exonuclease and an endonuclease. An exonuclease chews up an RNA or a nucleic acid from the end, and an endonuclease cuts it in the center. And in eukaryotes, like human cells, most basal RNA is happening only from the ends. So this enables the cell to regulate when an RNA is going to die by shortening its ends. And the major exonuclease that helps in that process is XRN1. And so it's a pretty efficient, fast exonuclease. And so these viruses, and we've shown that it's not just KSHV, but actually a number of viruses that use this sort of overt strategy where they encode an endonuclease, which enables the virus actually to short circuit the RNA decay pathway. So instead of having to wait for gradual shortening of a poly A tail or decapping, which tend to be the rate-limiting steps of RNA degradation, the virus can just short-circuit that by chopping an RNA in the middle, but then feed into the rapid 5' to 3' decay pathway that's driven by XRN1. And then maybe it's producing products that don't look all that different from what's being degraded in a cell.
So why isn't SOX cleaving enough, though? Or is it just a default pathway that once you have an end exposed, it gets chewed up by XRN1?
Why isn't it
so
enough?
Let's say, cutting an mRNA in the middle should be enough to inactivate it. Why do you have to have XRN coming in?
Yeah. That's something that we're in the process of exploring. So you're right, it pulls it out of the translational pool once you cleave it in the middle. But we're starting to think about why actual degradation of those RNA fragments is important, and we think that it probably is important. One of the reasons why it might be important has to do with some of the downstream consequences of this widespread RNA degradation. So this is something that we think a lot about. If a virus is coming into a cell and trashing most of the messenger RNA there, how does a cell sense that? And we assume it senses it. And then what are some of the consequences that happen from these falling messenger RNA levels? And one of the consequences that we've sort of been following up on is that a protein called poly-A binding protein, which is normally bound to the 3' ends of messenger RNAs, gets relocalized to the nucleus. And as it turns out, that process is probably dependent on the actual RNA degradation by exorin-1. And the reason that's important is that poly-A binding protein is a protein that's an important messenger RNA stabilizer and translation factor in the cytoplasm. So you can think of it as a general enhancer of gene expression there. But when it goes into the nucleus, we found it mediates the opposite outcome, where it's sort of, you can think of it as a repressor of gene expression, and that's because it blocks messenger RNA export and induces sort of a RNA processing abnormality. So by inducing degradation of RNAs instead of just their cleavage, you're going to have these secondary effects where you affect nuclear processes in addition to cytoplasmic processes.
Right. So otherwise the cell could just crank out more of the mRNAs.
Sure. So now you can't per se recover simply by ramping up transcription, for example.
Right. So where does the cleavage first by SOX occur in the cell? Do you know?
Cytoplasm.
It's cytoplasm, and are these on polysomes, or are they just RNAs floating around in the cell?
Well, cleavage can occur on polysomes, but we think it doesn't have to occur on polysomes. The RNAs have to be translationally competent, but they don't actually have to be translating. So binding of 40S ribosomal subunit is not even required. So it's a very early stage if translation is involved at all. But if you make an RNA translationally incompetent, it can't be recognized anymore. So there's some feature of messenger RNAs that allow them to be targeted.
And is it, I guess it's all messenger RNAs in the cell, and then, so other viral RNAs are different, but they don't have poly-A, or some of them don't even have caps. Do you know if that makes a difference for the degradation?
Well,
probably haven't looked at it, I would guess.
Yeah, we've actually thought about ways of trying to look at that. And what we know is that the tricky thing is that for engineering RNAs, for example, without poly-A tails in a cell, that's going to kill their translatability. And so that's one of the ways that we actually showed that translational competence was important. One of the experiments that we've thought about doing, which you're kind of alluding to, but we haven't for sort of technical reasons, is looking at, co-infection with viruses like coronaviruses or dengue. We had talked to some people in your lab about that, Eva, to see whether or not, what are the actual, physical components of a messenger RNA that's critical.
There are you published a paper on some other viruses that use XRN1 also, right?
Yep.
To take advantage of that pathway.
That's right. SARS coronavirus, and some other herpes viruses too.
Right. So can we go back to the IL-6 story? I didn't ask the other question I wanted. You said it's refractory to SOX cleavage, right?
Yes.
So what's involved in that?
Yeah, so that what's involved in the protection is an element in the 3' untranslated region of interleukin-6 messenger RNA. So it turns out cytokine RNAs are pretty labile. They get turned over rapidly by the cell, and that's because the cell wants to be able to turn them on and then turn them off really fast. And so this is one of those RNAs that has a lot of regulatory elements in its 3' untranslated region, or 3' UTR, that are going to help turn off that RNA. And interestingly, we found that this region of the IL-6 RNA that is highly regulated is the same region that is important for conferring protection against SOX cleavage. So we can actually just take a small segment of the RNA, like 200 nucleotides, fuse that onto a completely heterologous RNA, and confer protection.
And presumably that sequence binds something.
That's right. We think that it's through these RNA binding proteins that are complexing with that region, and we've identified a couple of them, but we think that there are a lot of other ones as well.
And is that just one way of doing this? You think there are other mechanisms as well?
Yeah, I would guess that there are probably a number of mechanisms, and we've hit on one of them. One of the reasons that we get excited about this sort of thing is that we think that these viral proteins are kind of offering they're great tools to figure out how specialized processes or very generalized processes in a cell work. And so by, if there are specialized mechanisms of decay, which we think that there are for certain RNAs like IL-6, then if we can figure out how the virus is manipulating that specialized decay pathway, then we can learn something about how it's normally working in our cells. So this is one mechanism. I would bet that there are others out there.
So other than IL-6, are there other majorly protected mRNAs that you know
There
of?
are other RNAs. That's the only one where we've really characterized how it escapes.
In one of your papers, you mentioned that the protection of, I think it was viral mRNAs, involves one of the viral proteins, or 57. Is that right? Do I have that right?
OR57 is a protein, so this is not a protein that is work done by us, but others in the field that can bind viral RNAs, and the idea is that it helps stabilize viral RNAs. But whether or not how it interacts with SOX in a cell functionally hasn't been explored yet.
Okay. So is there anything else about this aspect of decay that we missed that we should talk about?
How many hours do you have?
About 10 minutes.
I guess, I think we covered a lot of it, but I just wanted to emphasize, one of the things that I think is interesting and compelling about this strategy of decay are the number of different viral proteins that we're finding that hit upon the same strategy. So when you find something over and over and over again, that's a pretty important clue that it's important. And well, at least that's what we tell ourselves. That's what I keep telling my students. So this idea of, the overt strategy being similar in, the DNA viruses that we're looking at, but also RNA viruses as well. This idea that there are downstream consequences of that we're seeing in many different types of viruses. And from our work and others, in addition to herpes viruses, the coronaviruses, and now some of the Bunya viruses as well, with this poly-A binding protein relocalization and RNA export defects. So a lot of these viruses are interacting with these gene expression machinery in the cell in a kind of a similar way. They're tapping into the same basal machinery. They're leading to the same downstream responses. And some of these responses are happening in cells even in the absence of infection. So things like the poly-A binding protein nuclear relocalization phenotype. That's something that had been observed originally during certain types of cell stress, like heat shock, outside of a viral infection, but nobody knew what it was doing there. It was just sort of this observation that it can go into the nucleus. And so using viruses to figure out, well, how is this phenotype actually influencing gene expression in the cell, and how are viruses maybe using that to their own benefit, is something that I get really excited about.
Viruses point the way, right?
Yeah, that's
Always.
right.
They do.
They're like a
reveal
beacon of
things.
light.
So let's spend just 5 minutes on another aspect of your work, which you published in January. This is Lisa's work.
You are.
She wants us to give it 5 minutes, and she was so instrumental in getting us here. This is these upstream open reading frames.
Yeah.
And I teach my virology classes about the UORFs in CMV.
Yep.
I've done that for years, and how they're so close, and you can get termination and then reinitiation. So give us the elevator summary of that.
The elevator summary. So another thing that we think about is, how is it that viruses and people like you who are RNA virologists have been thinking about this for a long time, how do they get their genes expressed in a human cell when often viral RNAs don't necessarily look the same as a host RNA? They've got to use the host gene expression machinery, the translation machinery for sure, but they might not look the same. So what tricks do they have to employ to get their RNAs made? And so for things like herpes viruses, people don't maybe spend as much time thinking about that because their RNAs look very similar to a host RNA, right? They're made by, RNA polymerase II, they're capped, they're polyadenylated, they're made in the nucleus, sometimes they're spliced. And so you think, okay, well, it doesn't have to go through as many hurdles as something like an RNA virus does. But as it turns out, there are examples of RNAs in herpes viruses which still don't conform to what a eukaryotic messenger RNA looks like. And for example, the locus that Lisa works on is one where you have what's called a polycistronic RNA. So you have more than one gene for one messenger RNA. And all those microbial biologists out there, of course, this is a super common thing in bacteria, but it's pretty rare in a eukaryote. And so how do you get a gene that's at the 3' end of an RNA made when our ribosomes are just going to fall off before they get to that 3' gene? And so this is one of the things that we were looking at for a locus in KSHV, and we found that they use what's called a UORF, so a short upstream open reading frame. These are really common in eukaryotes. You can think of them usually as a translation speed bump. They're found in the 5' untranslated region of an RNA, and the thinking is that the ribosome will engage in these upstream open reading frames, read them, and that sort of slows them down, and then only a fraction of the ribosomes will actually engage at the authentic AUG of the major ORF. And so used all the time in eukaryotes, and in DNA viruses like CMV, they're all over the place too. What we found is that there is an example in KSHV of a UORF that instead of serving as sort of a translational speed bump, because of its sort of clever positioning, actually enables translation of this downstream gene, the second gene on the polycistronic RNA. So it's sort of an unusual repurposing of this common cellular strategy, which is another thing that viruses tend to be really good at.
So in this case, are they overlapping?
Yeah, so the UORF overlaps with the start sequence of the 5' gene. So if that UORF is recognized, the 5' gene is not, and the ribosomes can actually scan through the whole 5' gene and get to the 3' one instead, with a remarkable level of efficiency, actually. So that downstream gene is expressed much more robustly than the upstream gene, even though you would never predict that from the Kozak consensus sequence.
And how far are they? How is that downstream gene that's expressed better when there's the UORF? How far is that?
I don't know, 300-ish
That's a lot.
nucleotides. It's a pretty long range. Thanks for that cheat sheet over there. Lisa's like signing it to me. Don't get it wrong. That's my thesis.
300 is a long ways. I mean,
it's a pretty long ways.
You think it's just scanning, or is there some other kind of handoff involved?
Yeah. At this point, we think it's scanning.
Okay. Yeah, that's a cool story. I thought we should mention that. And the last thing I want to point out is polio is cool because you're talking about RNA viruses. It's got this protein at the end, which is weird. But then we think it's taken off before translation.
Yeah.
We're not actually, the ribosomal RNAs don't have VPG on them, which would leave you with a 5' phosphate, which says that these exos should be chewing it up, right?
Yep.
And so there has to be some inhibition, right? I got it right? Yeah, very good. All right, I have one more question for you. If you could talk to anybody who ever lived, who would it be?
Bruce Springsteen?
Really? Okay.
Seriously.
Sure, no problem. He's still alive.
I mean, not that there's not many fabulous scientists out there, but everybody's got idols, right?
Yeah. Okay. We were doing this the other day during a podcast, and I came up with like Louis Pasteur. Can you imagine sitting him down here with today's science? I wonder if he would get it, or if he would have the most amazing questions because he was a smart guy.
Yeah.
So that would be pretty cool to do. So he was one, and Ben Franklin would be another.
Now you're making me feel like I should have come up with a more intelligent answer to that question.
It's okay. They liked it. It's no problem. All right, Eva.
Yes.
Where are you from?
New York.
Really? You're born in New York City?
Oh, yeah.
Wow, I didn't know that. Never come and visit us.
Oh, I get there a lot.
Yeah?
Can't live without it.
You should come by and do a podcast sometime. I know we're doing one now, but we could talk even
Pick
more.
another. We can pick another topic. Yeah.
And where'd you go to college?
I was at Harvard undergrad, and then I did my PhD actually here in molecular and cell biology.
Berkeley.
Surprisingly, yes.
And a postdoc?
At UCSF.
No.
Kind of, actually, I won't say in Don Gannam's lab, but that's where I crashed his lab meetings and Raul Andino's to learn virology, because I have kind of a checkered history, and unlike the virologist, like all the way, from the get-go, it's been a circuitous path.
When did you get interested in dengue?
Well, like I said, I have this kind of science track, and then I have another track, which the mission was to develop scientific capacity in the developing world, and so I was kind of doing them both at the same time. And so while I was doing my PhD in yeast genetics, I was also running all over Central and South America trying to bring tools to places that barely had running water and electricity, and prove that you could actually use molecular biology in a way to do molecular and diagnostics and epidemiology and such. And everywhere I went, I would say, so what are your priority diseases? And it was tuberculosis and dengue. And so first I had to figure out how to spell it. Then I had to figure out, to my shock and horror, that it was a virus. Because I thought I was like a yeast geneticist. I thought, okay, this is so cool. Now I'm going to take this eukaryotic system and I'll work in. I was actually supposed to do my postdoc with Stan Falco and kind of move it into the bacterial kind of molecular genetic system. And then with Leishmania, I started working. So I thought that was just when the tools of molecular parasitology were coming on board. And I was so excited about that. But then life veered, and I ended up getting written up in Science in this profile about doing this kind of work, and at that point there wasn't nothing called global health. There was no words for this at the time, but there was a lot of interest, and here and there and everywhere, and I was kind of onto something, and so I just did it and ended up running around Latin America, and that's where I found out what an enormous public health problem dengue was. And what was interesting is that as the more I looked at it, it was like a black hole. I mean, there's so many interesting questions about the public health aspect and the clinical aspect and the epidemiological aspect, but there's a huge number of scientific questions, from translational control, which we worked on, as well as the immunology and correlates of protection, as well as the immunopathogenesis. So it was just kind of everywhere you look, there were questions to be answered.
Where did the desire to bring science elsewhere come from? Can you pinpoint that?
Just naively, idealistically, I wanted to do something in my life that could change the world for the better, and I also adore science. I love the scientific process. I'm fascinated by cells. In a way, I see the cell as kind of a model for human society because there's this great energy, efficiency, and then the cell and the organism all work together for the greater good. And so so I was kind of fascinated both at the molecular level as well as almost at a, metaphysical level, or at a philosophical level. And so I love biology and biochemistry, but I wanted to also be able to see an impact with that. And so I tried to kind of invent my way forward and
I've
make that happen.
I've read a bunch of your papers, and a number of them are involving studies in Nicaragua. Mm-hmm. And I noticed in the discussion, besides summarizing the science, you always say, and this shows that you can bring these techniques elsewhere and rely on them and trust the data and so forth. So that's a big part of it, right? Mm-hmm. I think that's neat. All right, let's briefly go over the dengue pathogenesis. So it's a mosquito-borne viral disease. Mm-hmm. Certain mosquitoes carry the virus, right?
Right.
Aedes aegypti and albopictus.
That's right.
Does the virus replicate in the mosquito?
Yes.
Yes, so that amplifies. So it picks it up from a human.
Yes.
It replicates, and then it transmits to another. And there's no other host as far as we know.
Right.
Just humans.
Yeah, there's a sylvatic cycle with primates, with monkeys.
Okay.
But
And in the U.S., we have A. albopictus, but we don't have much dengue, right?
Well, actually, there was a huge amount of dengue. So Benjamin Rush in the 1700s, et cetera. And then there's a lot of dengue along the whole eastern seaboard, along with yellow fever.
Right.
And so with the increasing socioeconomic status and draining swamps and kind of more air conditioning and screened windows, this kind of stuff, that really made a dent on vector-borne diseases. And so a lot of the effort was on controlling malaria and then controlling yellow fever. But and yellow fever is actually transmitted by the same Aedes aegypti mosquito that dengue hitches a ride with. And so as those mosquitoes were controlled, then dengue went away. along with yellow fever. But what's interesting is that there was a Pan-American wide campaign to eradicate, never use that word, eradicate Aedes aegypti, which was quite successful for a while until actually the US stopped paying into the fund, and then that stopped. And that was in like the 50s and 60s. And so there was a period where Aedes aegypti was wiped out of the United States and a lot of Latin America. But when the fund dried up, then it was reinfested with, and then not only with Aedes aegypti, but also albopictus. And so albopictus is what came back in. Actually, interestingly, albopictus re-entered the United States on a boat, a ship that docked in New Orleans in the 70s, and had on it used tires, and the tires was water, and it could come from Southeast Asia. So in the water were larvae, and then that spread throughout the Southeast United States. In fact, now dengue is endemic again in the United States, in the Key West and Florida. And then there's been several epidemics in Hawaii and
Texas also?
Texas, along the Texan border, yes. And a lot of those are imported cases, or people who actually are going to Mexico to seek health care because it's cheaper and coming back.
Okay.
And so
So there are four serotypes, right? And you get, let's say you get bitten by a mosquito the first time. What happens to you?
You can get really sick, like I did. No, so the most of the times where did you I know of which I speak. Yeah, so a lot of these are asymptomatic infections or inapparent infections, or you get primary dengue, which always bugs me. No offense to flu, because we actually work on flu, but it always bugs me when they say flu-like symptoms, because for me it's like it's called la quebradora. It's breakbone fever, and you feel it. It's intense. You know, you're wiped out, and you have these incredible pains in bones and muscle, etc. And you don't forget that.
That's what you had.
That's what I had. But it's called classic dengue. You can also have a more mild, undifferentiated fever. And we found actually in kids, in one of our cohorts studying to get out with it, a full 25% of laboratory-confirmed dengue actually don't comply with the WHO, World Health Organization case definition. So, because potentially, and mostly in young children, because potentially they can't say, "Mommy, I have myalgia," So, but definitely there's a range, and you can get the more severe form, which I'm sure you'll ask about, but that just generally happens with a second infection with a different serotype.
Okay, so a fraction of the primaries are severe, but not anywhere near. If you get a second infection with a second serotype, then that incidence. You could also still get the primary type of.
So the deal with the secondary infection is not at all that every time you get a secondary infection you're going to get the more severe disease. But the flip is true, where the most of the severe disease happens in secondary infections, and hence the idea of an immunopathogenesis, where it actually has to do with your response to the first. And this really scary thing about the severe disease with dengue. So it's an, unlike you, my dear, this is an acute illness, and so it's over and done with in 5 days, 5-6 days. So in a way you would think, okay, cool, 5-6 days. But the problem is that it's also very quick. And so in this more severe disease, which actually is really a vascular leakage phenomenon, which has to do with a cytokine storm that ends up working on the capillary endothelium and separating tight junctions and allowing fluid leakage out of the circulatory system, leading to what's called hypovolemic shock. Death can be very, very quick. And so 24 to 48 hours, you have a completely healthy kid, and then they just shock and die. And it's very frightening. There's no specific therapy right now, no treatment or no vaccine, so you rely on supportive care. But that means that you actually need to see it coming, and the see it coming is only a period of a couple days. So it's quick, but it also is much more dangerous. And you imagine, actually there was a Nature paper that finally came out, we've been wanting to cite this forever, but finally came out this week, where it's actually about 100 million cases a year and 400 million infections. So it's triple what the WHO has been estimating. And so it's a huge burden, and then some people are at risk for this very frightening severe disease.
So the severe disease, as I understand it by reading the literature, is antibody-dependent enhancement and possibly also T cells involved, right? So what is the antibody-dependent enhancement about?
So that has been an in vitro phenomenon, which actually through our work and others in mouse models, we can actually show that this actually can happen in vivo, and then it's thought epidemiologically to be true through the work of several groups in infants that have a very high incidence of severe disease. And it's thought to be due to antibodies which are transferred from the mother who's dengue positive, and will transfer antibodies that then get catabolized until they're no longer neutralizing. And before they're completely gone, there's this enhancement period of sub-neutralizing antibodies. And so what we did to prove this, because you can do this in vitro, where you just have a cell line that has FC receptors, and then the idea is that you have a dengue with an antibody from a different serotype, which is not neutralizing it, but through the FC portion of that viral immune complex can pull the live virus into a target cell, because actually, like what Britt was talking about, in this case, it's not B cells, but it's actually macrophages and monocytes and dendritic cells, which are the target of dengue. And these are all FC receptor-bearing cells, and so you actually end up essentially opsonizing or increasing your infection to your target cell.
And that's bad.
And that's bad. So what we were able to show in a mouse model was that we actually have a mouse model that can reproduce a lot of the features actually that you see in human dengue, like the vascular leak and low platelets and high levels of TNF alpha and IL-6 actually in 10. But what we could do is have a sublethal infection. But if we take that sublethal infection and prior to that we treat the animals with an antibody, a polyclonal mix against a different serotype, or even a monoclonal to a different serotype, then we actually end up with a completely lethal disease. And so that's enabled us to then get at the mechanism of what's causing that.
So it's weird because there are many other viruses for which we have multiple serotypes. Polio, there are 3 serotypes, but you don't ever see antibody-dependent enhancement. Why do we see it with dengue?
I think part of it has to do with the tropism, because this is actually, you're talking polio and others like West Nile, these are more neurotropic, and so you're not in the same system of the periphery and those types of cells, which is where this whole story plays out. And again, these are cells which then can secrete cytokines, and the cytokines are what trigger, in theory, this is all hand-waving, this cytokine storm, and hence this leakage phenomenon.
Yeah, polio I view as whenever it gets in the CNS, it's accidental. Most of the time it's in our gut, it's not doing anything, and there's no chance to get antibody enhancement there, so maybe that's part of it. Mm-hmm. So you published this neat paper recently where you treat people with a monoclonal, or I'm not sure if it was people or it was mice and cells in culture, with the idea of competing for the FC binding. Right.
So what we were trying to do is figure out, now that we can understand at the mechanistic level, both in vitro and in vivo, what is causing this, can we then use that to interfere and develop some kind of therapy? And so we already used the mouse model to do a number of preclinical testing of a variety of different potential antivirals. But the idea here was to see, well, if you know that it's necessary that there's an interaction between this viral immune complex and the FC receptor, if we make, and it turned out to be just a single point mutant in the FC region that obliterates its interaction with the FC receptor, do we knock out ADE in vitro? Yes. And in vivo? Yes. And then we said, okay, well now what if we do this therapeutically? So we actually have a lethal infection and we come in after the fact with this monoclonal antibody, which can no longer interact with the FC receptor. Then can we actually override, essentially dominantly? Right? And in fact, we can. 24 to even 48 hours later, we can come in and completely obliterate the lethal phenotype. So 100% protection of these mice. And so then we started saying, well, what's the mechanism of this? And so we started looking at a number of monoclonals, and if you have just a straight lethal infection that has just a high viral dose that leads to the kind of vascular leak, early death syndrome, then you can protect them with any of the monoclonals that were highly neutralizing. But if you have an antibody-enhanced infection, then it turns out that just being neutralizing wasn't enough. And it turns out that what you needed to do was to actually be able to, we think, essentially compete with or displace the enhancing antibodies that were present in the serum, and then be able to neutralize. So there are actually 2 elements. And we were able to actually start picking that apart and see which are the epitopes which are being addressed. And there's some kind of neat, interesting stories about where those lie on the virion itself, and et cetera. And
so if competing in this way is enough to get rid of serious disease, does that imply that there isn't a T-cell contribution to it, a cytokine storm type?
No, because essentially what that's doing is the thought is that you have the infection, viral infection of the cells, and then that triggers this cytokine storm through the monocyte macrophages secreting cytokines and then activating T cells as well. So there probably still is. It's actually very, very controversial right now. And there's a number of actually human trials as well as mouse work as well as in vitro. So there's a lot going on in this particular area right now about what the role of T cells is. In fact, there's a paper that came out this week in PNAS that shows that there is this this theory of original antigenic sin where you actually have more of a response to your primary infection in T cells, and we've shown this in B cells. But in fact, this actually might not be bad. It actually, might not. It might confer protection instead. But I think that the idea is that if you can prevent the initial viral amplification, then you might not trigger as much this more severe disease. But it's quite murky, actually, what the actual relation is between viral load. It's just kind of, we all assume that this is going to lead to the cytokine storm, but the actual molecular mechanism there's a couple theories out there and some papers in this area, but that's not really well established.
So are you thinking that your monoclonal approach might eventually get into people?
Potentially, we just wrote a grant saying potentially that might be an idea. And what we've done, it's interesting, we've been collaborating with several labs that we've been taking the PBMCs, the peripheral blood mononuclear cells, from our studies in Nicaragua, using them to make human monoclonal antibodies. And then collaborators at University of North Carolina have Dr. Ervin Silva has a traveler's clinic, and then we work with Jim Crow's lab, who's been making these human monoclonals. And then we actually have identified 3 classes of antibodies through all of this kind of work. Dr. Silva's lab and others have looked at, there's a type-specific response in primary infections that looks to be to a particular epitope and very, very, very potently neutralizing, but type-specific, so you would need 4. So we said, well, you could do something with 4 of these, but then there's another one that we just have isolated from one of our Nicaraguan secondary infections, which looks like an antibody which is actually cross-reactive and very neutralizing. So that's another one. And then there's a third class, which are these enhancement-suppressing ones. And so we're trying to see, and would this be reasonable? I mean, I think there really is a huge need for this moment where you have these kids that are suddenly very quickly going into this potential shock and death, and we need something. And so, and also there's of course travelers and whatnot who are going to areas and know in advance that they'll be exposed. And plus it's just, it's interesting to figure out something scientifically and then try and move it forward. But what we're really working on also is using all of our clinical and epidemiological studies in Nicaragua and to try and understand not the pathogenesis, but like the other 99%, essentially the protection. So we're looking at a much broader approach to immune correlates of protection using our natural infections in Nicaragua and then linking them with our colleagues who are developing live attenuated dengue vaccines. And that's what I'm really excited about these days.
Tell us about it.
Yeah, so I think there's a number of vaccines that are coming down the pipeline. The 3 front runners are live attenuated vaccines, but it's tricky because you have 4 different vaccines, and really we think of these as dengue viruses, but in fact, if you look phylogenetically, they're essentially equally distant as the viruses that get their own name and disease, like, for instance, yellow fever and West Nile and St. Louis encephalitis, and these are all flavies and they're in their own right, but they're actually just as it's just as distant as the 4 dengue viruses. It's just that the 4 dengue viruses can cause the same kind of disease, but really they're very different, so they actually interfere with each other. So it's very challenging to develop these, and of course they're RNA viruses, so they're constantly mutating. So and actually, if I have time, I'd love to tell about a story which is really looking at this question. But it's within each serotype, there's genotypes. Within these genotypes, there's clades, and all of that can make a difference both for pathogenesis as well as potentially for protection.
Is there, can I interject a quick question? For these tetravalent vaccines, is there a risk that if somebody develops a great immune response to 3 but not 4, you're at enhanced risk for getting? So, but
that's some of the points that we want to look at. And so what happened was that there's been a lot of work in developing vaccines, but what's really lagged behind it is the science around the vaccinology and understanding the immune response. And so we understand a little tiny bit about the pathogenesis and actually very little about protection. And so the idea here, so we have this really neat cohort, which is, we have 2 studies which are going on for essentially 10 to 15 years in the same hospital, in the same health center in Nicaragua. So one of them is a hospital-based study where children come in sick, and then we, of course, give them medical care and come up with a lot of different aspects through our research and clinical elements that can really help in terms of the treatment, et cetera. But then scientifically, we're looking at capturing the 3 acute phase samples and then a convalescent sample, and then 3, 6, 9, and 12, 18 months, so we can see what happens with the immune response. But we don't know anything about these kids before they come in. We just know that they were sick when they got there, and we have a certain amount of disease severity. But this has been going on in the same hospital for about 15 years. And so then we have another study, which is a cohort study, and this means that you enroll children healthy at the beginning, and this was in 2004, so about 3,800 kids, almost 4,000 children. And we're just following them and now it's in its 9th year. And so what's really amazing there is that a lot of the same kids have been there for many years, and so we can actually capture repeat infections. And what we do is we not only follow all of their febrile episodes and work them up for dengue, but importantly, we have a healthy blood sample taken every year. And so what that allows you to do is to look in those healthy samples every year and see if you have a 4-fold or greater increase in dengue antibodies. We know that kid was exposed to dengue, was infected. And we also have been following every single medical episode, so we can capture all the ones that were infected and got sick. But the cool thing is that we also have all these kids that were infected and did not get sick. And the even cooler thing is that we have the pre-infection sample. So now what we can do is take this pre-infection sample and try and then see what is it that can correlate with disease, no disease, which is exactly what the endpoint of the vaccine trials are. And then, of course, if we have severe disease, then we can ask that also. And actually, Molly Oinley, who's here in the audience, did a beautiful story looking at this increase in disease severity that we saw, and we said, well, what's going on? But it was in both of these 2 studies, so it wasn't just some arbitrary weirdness, some artifact, because it was 2 different studies, and there was something epidemiological happened, and she ended up being able to track it down to a change in the clade of the virus. It was all dengue 2, but it changed within it. But then we thought we had the answer, and then in the end it fell apart. And we said, well, okay, it's not just the genetics. What else is going on? Is it the immunity? So then we looked, and in fact, yes, there was an element of the waning immune response, and then we were able to essentially reconstruct the immune history of the children and find out that it was actually, the severity was a product of 2 dynamic processes that were going on through time. The change in the viral genetics and the change in the serotype-specific immunity of the children. So it was really interesting. So kids that were immune to dengue 1 previously were getting more severe disease with this particular clade 1, we called it, of dengue 2. And kids that were immune previously to dengue 3 were getting more severe disease with the other clade 2 and of dengue 2. And this is why it's so complex, because you actually need to be able to understand the whole history. And of course, put together a team of virology, immunology, epi, biostats, clinical. And when you say what gets you excited, that's what really gets me excited, is to be able to pull together teams from different disciplines and try and explain at a laboratory level what's really going on in human populations.
Are you also looking at the genetics of these people to see if there are any polymorphisms?
Yes, we are as well, and we have ongoing collaborations on several, actually, with different groups looking at the host genetics as well.
Now, for a number of viral diseases, when there's a severe infection, we know that innate immune genes are really important, and mutations can cause herpes encephalitis, for example, mutations in TLRs. So
yes, those are ongoing, actually.
Similar things are going. So do you think a vaccine is something that's going to happen, or is it hard to do?
Well, so what happened was that the first vaccine trial, it really was a phase 2B, and it was this in Thailand. They ended up getting a dengue 2 epidemic, which gave them enough cases to actually do an efficacy outcome. So it's a proof of concept efficacy trial. And this is in a tetravalent vaccine by Sanofi Pasteur, which is actually in the yellow fever 17D backbone. So there's been hundreds of millions of this vaccine, and so it's considered quite safe. And then within it is the the most important structural proteins, the PRM and the envelope protein of the 4 serotypes of dengue. And what happened is that they did this trial, and it actually failed endpoint. And so it did not protect against the dengue 2 epidemic that came in. And another piece was that the neutralizing assay that they were using actually also failed to predict this outcome.
Yeah, that's right. I heard that. They generated neutralizing antibodies in culture,
so the thing is, it's not necessarily, we just don't know whether that particular assay was, and so what we've been coming all these years saying is we need to understand you use our natural infections to understand what would be a best correlate of immunity to use in vaccine evaluation. And so that's exactly what we're doing. So we're developing a new neutralizing assay that looks at essentially human monocytic cells with the right receptors, Fc receptors, another important receptor for dengue, Nicaraguan viruses, the right maturation state of the virion, and actually try and make the most biologically relevant situation to measure neutralizing antibodies, maybe plus C1q, because complement can be important. But then not only stick with neutralizing antibodies, we're actually looking also at other antibody effector functions like antibody-dependent cytotoxicity, so ADCC, and a couple of other different assays that we can measure to see what can correlate, again, in our cohort with disease, no disease.
Yeah. Why aren't antivirals part of this solution?
They are, and so we also have a whole story going on with antivirals as well as the many others. And what we actually, some of our translational control work in the molecular virology aspect of the lab had led to some thoughts about what might be important for developing antivirals, also our work on replication. But right now we're kind of more using the mouse model for preclinical testing. And the cool thing is that we can look not only at at drugs or compounds that could knock down viral replication, which everyone thinks you should do, but again, it's a little murky how that gets from the high viral titer to the disease. But since we do have this vascular leak disease, which is associated with high, for instance, TNF-alpha, which is not very specific, but it is what's associated with this in human dengue, then we can actually also look at, and we're doing this right now, at certain molecules that can actually counteract the disease itself. And so we can look at the antiviral and the anti-disease, and so that's kind of what, and I think that honestly it's, everything. You're going to have to throw the book at this thing, and at this virus, and we'll have to look at antivirals, we'll have to look at vaccines, and then the last piece is we just finished a random controlled cluster trial in Nicaragua looking at a community-based evidence-derived approach to control dengue via the mosquito.
Right.
And in like a non-pesticide dependent manner.
Yeah, so there have been some releases in Australia on Wolbachia infected. Is that what you're thinking?
No, we actually, I'm working on that also, but from kind of viral dynamics point of view, but not on the actual, we're just collaborating with Scott O'Neill, who's been doing this at a test site in Colombia. But that's a very interesting story, which I can get into in a second. What we've been working on is really just simply controlling the mosquito population by getting rid of where the mosquito breeds. And the kind of problem with this mosquito, Aedes aegypti in particular, which is what really brings dengue through the epidemic cycle, is that it breeds in clean water around people's homes. And so it's difficult. The public health programs can target it, but they go into people's homes every X number of months, and that's not a very effective way, and it's very chemical dependent. And so it's been shown time and again that if you just get rid of, with the clean water where the mosquitoes are breeding at least once a week, and we say por si solo cada 8 días, once a week you get rid of where the mosquito's breeding, then you can actually break the life cycle. And so once people understand that it goes from the egg to the larva to the pupa to the live mosquito, and that you can cut this every 8 days, every week, then you actually control, so you bring down what's called entomological indices, so the amount of mosquito in the home, and then relate that to the infection rate for the kids, and
sometimes
then of course
low-tech solutions are very effective, right?
Yeah, and keeping the motivation going is how do you do this, and so now we're working on how could you move it into a government. Government kind of program, but it still has to, like nacer, it has to be born of the community, and so it's just very interesting about how to make be sustainable. And so that's kind of the place where we are right now.
What do you think about Wolbachia release, infected mosquito release? Is that feasible?
Yeah, I was kind of a little more eyebrow raised until I kind of cornered Scott O'Neill, who's the one who's behind all this. And I was concerned about kind of letting something loose into the environment, monarch butterflies. All of this, I'm not a big GMO fan, but this is actually, it's very interesting. It's actually a bacteria which is an endosymbiont to a lot of insects, and actually to nematodes as well, which is a fascinating story for the filarial protozoal diseases, but not the filarial diseases, the helminth areas. But what we also know is that this particular Wolbachia bacteria lives in a number of different insects, but it was mostly in Drosophila and others, and not in Aedes aegypti mosquitoes. So what Scott O'Neill did was to work for years and years to get this Wolbachia into Aedes aegypti, and what that does is it has a life-shortening effect, and since that can actually make the mosquito die earlier and therefore not have as much time to transmit, but the dengue, for instance. But it also has a particular effect against dengue, so there's kind of a double whammy. But of course, the strains that have more of an effect against dengue they also have a fitness disadvantage, so they don't penetrate the population as much. And so with all of that, you really have to make a big effort to get this mosquito with the Wolbachia into the population, and so it's not just like one will wander off and suddenly spread like crazy. I was thinking, especially for instance, like Singapore might want this, but Malaysia might not, and you have 250 migrant workers coming every day to Singapore. How do you stop that from spreading? So at this point, I think it's quite interesting, and there's a number of studies going on, and we're actually interested in the impact on the viral population dynamics and issues around resistance potentially, and what that's going to do.
So you didn't want to say the E word, but it sounds like this is eradicable when you have the right tools, right? Because there's no animal host, they're just 4 serotypes.
But it's the problem is that the mosquito vector and dengue, it's not only that it's adapted evolutionarily to the host and to the mosquito, but it's adapted to the ecology of human behavior. That's what I actually find most interesting, and also to the global politics and the socioeconomic. So it's got everything all mixed up. And so where this is targeting is mostly in urban areas, in tropical, subtropical regions where it's unplanned urbanization. And the trend right now globally is into huge levels of urbanization, and up to 80%, 90% of Latin America by 2050 is supposedly going to be urbanized, and it's all unplanned urbanization with poor waste and water management. So you just have this rife and breeding grounds, for Aedes aegypti, and that's why I think it's going to be incredibly hard to control.
Mm-hmm. Okay, I have one more question.
Don't ask me who I would want to talk to.
Yeah, who would you
No, I don't know. There's so many people.
You had so long to think about it.
I didn't. I was talking too much.
All right, I'll give you 5 minutes while I wrap up. How's
Okay.
that?
Oh, no, the pressure's on.
So this episode of TWiV will be found at twiv.tv, our website, and also on iTunes. And if you like what we do, one of the things you can do to help us is to go over to iTunes and leave a comment or rate the show with stars, and that keeps us very prominent there in the iTunes directory and gets more people to subscribe and learn about this very cool science that is virology. We always love to get your questions and comments. Send them to TWiV at twiv.tv. Britt Glaunsinger is in the Department of Plant and Microbial Biology here at the University of California, Berkeley. Thank you for joining us today.
Thanks very much for having me.
Did you have fun?
I certainly did.
All right, very good. Eva Harris is in the Division of Infectious Diseases and Vaccinology here at UC Berkeley. Thank you for joining us.
Thank you.
Did you come up with an answer?
No. But it would be in the arts, actually. I'm an avid reader. I have a number of authors who I really like, and also in the area of architecture. So that's actually even more so than science.
Well, that would be cool. Well, you got some time. You can still.
I'll write it. That'll be okay. When I'm in New York next,
all right,
I'll be prepared.
All right. Well, listen, when you are in New York and you have time, come up and we'll do another podcast, because it sounds to me like you have even more to talk about. And then you have to give me the answer, okay?
Yes.
Fair. But anyway,
promise.
Thank you for joining us today. I appreciate it. I'm Vincent Racaniello, and you can find me at virology.ws. I want to thank the Microbiology Graduate Students here for having us. Thank you so much. I really appreciate it. In particular, I want to thank Emma for corresponding with me and arranging everything. Thank you, Emma. And I want to thank Lisa and Zoe for picking me up at the airport last night. And we had a great ride on BART where we talked about, guess what? Viruses. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.